Hello, welcome to our interactive session number 2. So we are going to work and to dream about what is going to happen in 10 years or so, with an impact of molecular tumor board, what treatment solutions, how can we support clinical decisions, and how can our patient have an access to the latest innovations. So we have two speakers today on this session. Dr. Laura Oprea, who is working in R&D partnership manager, and she's based in Switzerland. And Tara Love, who is a medical director in molecular diagnosis and precision medicine for the Roche Information Solutions. They are going to give a speech together, and we will take the speech one after the other one, and we will then have a room for discussion and debate on those new tools and new approaches for our patients in the future. So I think we can start with our discussions and with our presentation. Sorry.
Hello everyone, my name is Laura Oprea. I'm the R&D Partnership Manager for RIS at Roche Diagnostics, and I'm very happy today to start this presentation along with my colleague Tara Love, MSA and RIS, with the title "Each Patient Needs Access to the Latest Innovation. Sharing is Caring." So today we can match a blood transfusion to a blood type, right? And now, what if matching a cancer cure to our genetic code was just as easy, just as standard? What if figuring out the right dose of a medicine was as simple as taking our temperature? What if cell therapy is as easy to produce as a pill? What if it's not only an option for end-stage oncohematology patients? What if at birth the umbilical cord blood is collected and represents our best heritage that our patients could leave us as a source for any potential cell therapy? Now imagine again, it is early morning of 28th of March 2013, that when looking at your smart mirror, it beeps a warning notification. Today we can match a blood transfusion to a blood type. And now, what if matching a cancer cure to our genetic code was just as easy, just as standard? What if figuring out the right dose of medicine was as simple as taking our temperature? What if cell therapy is as easy to produce as a pill? And what if it's not only an option for end-stage oncohematology patients? What if at birth the umbilical cord blood is collected and represents our best heritage that our parents could leave us as a source for any potential cell therapy personalized for each of us? Now imagine again, we are in 28th of March 2030, that when looking at your smart mirror, it beeps a warning notification that new mole on your chin should be checked by your dermatologist. That was to be expected. After all, your genetic test revealed that you possess a mutation in the CDKN2A gene. But you also get the recommendation to have your esophagus and stomach checked, as your connected smart scale registered a noticeable drop in your weight, and this mutation also carries an increased risk of gastrointestinal tract cancer. What if now we just come back to reality? I would like to spend a couple of minutes with you on the challenges, in a personalized oncology, and also to show that these challenges cannot be solved alone, right? And one of the major questions here is what and when a digital solution. To date, in personalized oncology, there are many challenges from different perspectives. And if we are having a closer look from the patient perspective, we see that access to precision treatments and trial is done case by case. 5 countries only perform reimbursement for such treatments. And these cases, the median time to treatment initiation is more than 3 weeks. In addition, rural areas has higher death cancer rate, and 60% more time is spent by rural patients versus urban area patients to see their doctor. This, of course, leads to inequitable outcomes even within the same country. If we are looking now turning from healthcare provider perspective, a recent survey run in 2019 among ASCO members shows that 51% of oncologists see lack of ability of finding suitable trials and reimbursement for the drug and genomic test as main barriers for precision oncology use. In addition, only in 2019, more than 12,000 clinical trials were initiated in Europe, and more than 6,000 clinical practice guidelines were published on PubMed since 2015. And in this context, physicians need to take decisions based mainly on experience, since they cannot take into account all the existing information. And I suppose this is normal. If we are looking now from payer perspective, the reimbursement from molecular guided treatment is often denied due to insufficient study evidence. On the top of that, COVID-19 pandemic impacted payers' priorities in terms of reimbursement, and they expect budget retraction from 15% in US to 25% in Italy and Spain, as pointed out by a recent survey in this country done this year. Trials face additional testing challenges, which in combination with the amount of available data lead to inefficient recruitment. For example, with few patients tested, the enrollment is below 5% due to limited referral of screen-failed patients. And this is not sufficient. Predictive biomarkers are associated with more than 60% of new therapeutics launched in 2018, and 40% of trials requires a biomarker stratification. If we are looking at all these challenges, we realize and we see logically that this cannot be solved by one stakeholder alone. During previous slides, we showed the role of evidence and the data in this, and how this evidence based on data hold the potential to fill the gap and bring some elements in front of those challenges that I mentioned before. However, not all the data is the same. And the philosophy, we have basically a philosophy of duality in real world data. We have a component of exploration and a component of exploitation in a real world data context. And we're using this way to structure the data needs for different purposes and point out the main challenges to extract the insights value for each of us. Now let's take a closer look to the clinical decision support using artificial intelligence and machine learning as underlying technology for algorithm training. And we see that the hybrid model emerges if we compare with the real-world evidence and translational research. For simplicity purposes, the real-world evidence in this situation doesn't make the difference between the diagnostic and the drug component. So why we have this duality, this hybrid model? This is due to data needs for algorithm training, which has to be different from data used for calibration purposes. For one is an exploratory approach, since we don't know which variables are improving the predictability of the model. And the other one is more exploitation approach, since at this moment, we should know what variable improves the predictability. The exploration component of algorithm training is very close to exploratory angle of translational research, where we don't know what variables are needed to discriminate between responders versus non-responders, for example. The exploitation component, on the other hand, is of the algorithm calibration is closer as a concept to the classical real-world evidence studies for market access, safety, for drugs and devices, where we know exactly what data variables we need to build a case for regulatory purposes mainly. Now, if we look from the angle of the way of extracting the value, and I have in mind the intellectual property, from one hand, we don't want to block the innovation, right? But on the other side, we don't want to block the freedom to operate. As you can see, there are other several challenges that I'll not develop in detail, like the transition from algorithm training to calibration, and also to demonstrate the medical validity of the AI-based software as a medical device products, and within the scope of medical device regulation, the new regulation at European level. Managing this duality is a challenge, as often the needs of both these activities are contradictory. And it is very clear at this stage that intelligent flexibility, that then rigid dogmas are essential for such a paradigm to be successful. And the close and transparent collaboration between the various stakeholders is necessary to avoid chaos. In addition, there might not be a single solution to this dilemma, and scenarios need to be developed and to be assessed and understand how it resonates with each of us. And last but not least, such an approach might fit in very well with the emerging pay-for-performance paradigms in oncology being adopted by payers. We've seen that despite enormous advancements, today's routine capabilities are not sufficient to allow patients rapid access to innovation. There are a few reasons to that. The therapy decision is monodimensional, one gene variant, one targeted therapy. Evidence suggests that a very low percentage of patients had a partial or a complete response. Now, the next main scientific question would be, can we draw additional insights from the unexploited data before even generating a new one, so we can draw the right portrait of a tumor and ultimately increase the number of responders? Well, I suppose we are still working on the question and try to find the answers. There are other reasons of this very low percentage of responders. Those reasons, there are incomplete understanding of the relationship between cancer phenotype and genotype, and last but not least, limited drug options and access to match the molecular abnormalities. In this direction, prominent technologies at different levels of standardization and development are or can be used to look beyond bulk genomic and advance our understanding of cancer biology. Some of them are sequencing, single-cell sequencing, like single-cell DNA sequencing, methylation-specific sequencing, or single-cell RNA. Assessing proteins via technologies like antibody and mass spectrometry-based technologies like CyTOF, or drug phenotypic analysis like pharmacoscopy or organoids approaches. However, the clinical utility of the technologies mentioned still needs to be demonstrated. Today, we are still lacking consensus on the clinical utility for broad molecular profiling and the correlation between the genotype and response. There are a few interventional medical device studies ongoing in oncology where a new technology beyond bulk DNA is assessed. And ultimately, the continuous challenge of acquiring the knowledge as it evolves on the cutting edge to understand tumor heterogeneity. Therefore, solutions are needed to foster proactive discovery, continuous learning, and inter-organizational benchmarking of molecular therapeutic insights that become routine and are accessible to patients. Basically, the real turning point will be unlocked when continuous learning from research will inform routine clinical care. But what solution are we talking here about? We've seen that the current solution are mostly intelligent knowledge-based solutions who organize the genomic data, but who do not take into account the patient context, with no ability to give recommendation at the point of care. Medical experts decide on the clinical actionability of the data, and connections are done in a subjective manner based on their experience and knowledge, with no possibility to go back on the reasoning and understand why some decision have been taken in a way or another. Ideally, tomorrow digital solutions will be able to rank the actionability of combined alteration with a patient context with all the available multimodal data. In this way, treatment options become meaningful, objective, and transparent by learning from continuous new evidence, including from novel profiling techniques, as mentioned previously, and holistic view of longitudinal patient journey, starting with diagnostic, or even before, if we think to hereditary nature of some cancers. To achieve this, however, few steps are required. The key one is to be able to conduct molecular tumor boards, to prospectively structure at source the output data and further exporting and integrating it in a compliant manner and following all the ethical rules into a sustainable and continuous learning loop to assure the transition from rule-based towards a clinical decision support. And this is where my colleague Tara Love will present you the maturity model, the precision oncology maturity model. Tara, the floor is yours.
Good afternoon. My name is Tara Love, and I'm a clinical geneticist on the medical and scientific affairs team for Roche Information Solutions. I'm really excited to be here talking to you in this session, and I'd like to continue on with the spirit of what Laura was mentioning in her presentation around molecular tumor boards really being the enabler of data sharing. And I'd like to actually dive a bit deeper into the molecular tumor board topic, where we can specifically begin to get a better understanding of molecular tumor boards and how institutions are really adopting precision oncology decision-making around the globe. We would not all be sitting here today if we didn't all fundamentally recognize that healthcare providers are running up against challenges when trying to implement and execute molecular tumor boards. And I'll just highlight a couple of these things. You know, we all know that there really are no quality requirements or guidelines published to date that help institutions have best practices and standardization around implementation and execution of molecular tumor boards. And let's face it, interpretation of genomic data, you know, even if you do it for years, and translating those data into diagnostic and therapeutic insights are a pretty daunting task for us, even for the brightest molecular experts. And this gets confounded by even the complexities of trying to apply disparate and dispersed molecular evidence to the patient's clinical picture. And so even if we do all those things right, the recommendations that a molecular tumor board makes are often stifled, no matter which country, which region, by access and reimbursement challenges. So I would like to show you a bit about a maturity model I have been developing to begin to understand sort of where these challenges are similar or different across different molecular tumor boards around the globe. So I'm going to walk you through the current iteration of something I'm developing right now. And what I'm showing you here is the makings of a house. And so I'd like to start with the foundational blocks of the house that we're about to build. And they're represented by pillars. And so we have first the evidence pillar. We then have a technology pillar, a pillar around discussion and attendee profile, and another around access and advocacy, which is enabled by the molecular tumor board. And so what you can see here is there are different levels of maturity from 1 to 5 as you go from bottom of the pillar to the top of the pillar. So for example, in the evidence pillar, you might say that the least level of maturity or the lowest level of maturity could be that individual knowledge is used for clinical decision making. But as you layer in more pieces of evidence, you then have the ability to make more rich and more mature clinical decisions. And it might be that even at the highest level of maturity in this evidence block, real world evidence is used for clinical decision making. In the technology pillar, you can see sort of a similar progression where you might start with basic tools and an unstructured approach like Google searching and other things. And as you layer in more technologies, the highest level of maturity could conceptually be this idea that you could comprehensively look at multimodal data and aggregate it and generate insights from that. And you might even do AI predictions on real world evidence. In the discussion and attendee pillar, you can see a progression from potentially like ad hoc discussion, where you then might even have a dedicated molecular multidisciplinary team where you have layered in diverse molecular expertise. And that maturity could reach the highest level, say if you're a core MTB service provider, and maybe you've implemented quality and process improvements into your program. And the fourth pillar, I'll just touch on the most mature level, is that this idea that a molecular tumor board could be an influencer or driver of policy, for example, reimbursement. So as you reach a certain threshold of maturity levels in this model I'm developing, you form a rooftop. And Laura alluded to some of these advanced capabilities. If you're at a certain maturity level, the idea is that your system might have, proactive discovery baked into it, or could be a continuous learning system, or certainly could foster the sharing of data across different organizations. So you can start to see how levels of maturity here could be measured inside an institution. But admittedly, as you look at this, clearly it's quite high level. So we really can't assess maturity properly unless we're really drilling down into each one of these boxes. And that's happening right now. And I'm going to show you an example of sort of how I'm approaching the drill down here. So I'm drilling down each pillar to give us a richer maturity assessment. And I've kind of broken it down into two things, into vertical expansion and to horizontal expansion. And so for vertical expansion, I'm really talking about taking one of these boxes and doing a deeper definition of the individual blocks. And for horizontal expansion, I'm really talking about taking and translating the entire block or the pillar into a sub-pillar or dependent sub-pillars, plural. So in the vertical expansion case here, I've chosen to kind of highlight real-world evidence and how we could drill down deeper into this block. And you can see, you could start to categorize and say, well, I want to categorize by sources of real-world data that are then translated to real-world evidence. And these might be EHR data, insurance claim data, wearable device data. You might want to look at types of real-world data, outcomes data, for example. And you can drill even deeper and ask the question, well, what types of outcomes data do we care about to make our clinical decisions? So for instance, survival curves or treatment response rates. So I'm just trying to illustrate here, we can go several levels deeper. And I believe that this is critical to get granular in order to really understand an institution's molecular tumor board maturity. In the horizontal expansion example, it's clear that evidence is required, obviously, for you to do actionability classifications. And so you can't really get an AMP or an SCAT tier without evidence. And so this is just a prime example of sort of how you could have dependencies expanding out in the horizontal direction. So what do I really expect to get out of all of this? I think it's important to kind of highlight that assessing molecular tumor board maturity across the globe really will increase our understanding of how different organizations are operating and get us closer to best practices and standards definition. For Roche and for others, it can ensure, evolving products, clinical decision support products, and other things really are meeting our unmet customer needs. And thirdly, we can really help our customers in their transformation towards precision oncology services by helping them measure sort of where they're at and identifying areas, 1 or 2 areas, for example, where maturity could be increased. And how could we potentially help them in that regard to co-create the most appropriate solution to meet their needs. But first things first, the maturity model I developed obviously has some assumptions baked into it, and we need to test those assumptions. So to do this, I developed a companion scientific survey with input from experts from around the world to really get at the ins and outs of molecular tumor board operations and patients. And you can see the survey sections here, molecular testing, molecular data interpretation, and the tools that molecular tumor boards are using. And importantly, how molecular tumor boards are really measuring success. So if you'd like to share your experience as advanced centers of excellence to contribute to achieving best practices and standardization, I invite you to take this survey. And in return, I can share with you insights of the survey, not only from the attendees here, but from other regions of the world in which I am launching the survey. So if you are interested in taking this, I ask you to please text us at the number on the screen. And finally, I'd be happy to entertain questions on the survey and maturity model during the Q&A session. Thank you.
Thank you very much, Tara. I'm looking forward mainly on the pillars of evidence and technology and see how we can advance these 2 pillars together. As we've seen so far, instead of a conclusion, I would like to put these ideas and these topics, working directions that we are having from our end. We see that it's mandatory to systematically build the evidence into this continuing learning digital tool. Being able to learn from every decision taken during molecular tumor board could bring us closer to the vision from the introduction, and where, of course, ethical secondary data usage is instrumental, and we're going to look into the detail of that. Additional technologies like multimodalities to inform therapy choices beyond guidelines to improve outcomes, or other technologies to make data fair like NLP, advanced analytics using artificial intelligence and machine learning will enable what we have just started today. Thank you so much for your attention, and I'm very happy to be here.
Great. That was an excellent session. Thank you very much, both of you. It was very, very interesting and so promising. So we have 2 questions to start. So first question, based on the fact that molecular alteration clinically validated are most of the time included in the organ entity tumor board, what do you imagine as a molecular tumor board? Meaning, does it part of a regular tumor board, or is it separated?
Well, the way I imagine it, and the way I very much view it, is that it is a dedicated topic meeting specifically to go through the molecular alterations, not so much about the broader multidisciplinary team. In my view, it is a dedicated topic meeting, and it should be time spent to really go through those variants that are complex ones of unknown significance, or in combination with other variants. Not so much the smoking gun variants that are in the clinical practice guidelines, but in the way I see it, it's more of a forum to go through, those variants that need a deeper dive and a deeper dive discussion. Hopefully that makes sense.
Okay. I would say that in some tumor types today, like lung, it's complicated to separate the tumor board from the molecular tumor board, because most of half of the patient will have molecular alterations. So it's a balance that is different, especially if we have a specific driver. It's more in terms of organization, how do you split the patient from one board to another board, or but it's more when we have not classical alterations or concomitant alterations also. What will be your strategy if you have targeted two different targeted therapy or immunotherapy and targeted therapy eligibility. And here again, probably the other tools will help us a lot by gaining experience from other colleagues, because today we have sometimes some clinical reports, small case reports, but we don't have guidelines for all those situations that occurs more, all those situations that occur more and more right now that we are testing the people. We have another question, which is, because of the aim of a tumor board, is to define a therapeutic strategy for the patient. What do we do if there is no drug available, even if you have identified a molecular alteration? So what is the value for you on that?
That's an excellent question. Do you want me to take this, Laura?
Mm-hmm.
Okay.
Maybe Laura will have also
so
something to say.
I can have it. Yeah. So
yeah, go ahead, and then I can jump in or whatever.
I can start. Absolutely, I can start. Depends on depending on the abnormality, right? I mean, it's if there is any therapeutical options which is outside that particular indication, that it can be considered innovation. But depending on the context, depending on the country situation, that patient could have access to it in a certain circumstances. There is another way if there are any clinical trials which are ongoing on that abnormalities as well. So at least there are two ways of for that particular patient to access innovation. If there is no possibilities, then I do agree with reach kind of a sort of and this is to the team to take a decision for that situation. Tara, anything to add on this?
Yeah, and I think that the more we institutions adopt dedicated molecular tumor boards, the more the more we have this adoption, the more possible standardization and best practices can be defined, and that I think is going to give us a virtuous cycle. I mean, maybe I'm thinking too idealistic, but I do believe that if we get to a point where we have practice guidelines for molecular tumor boards, that's going to help us get access to those treatments that are not on label, and combination therapies that are emerging out of the literature and other places like that. So that's all I wanted to add.
So I can add the French perspective is that it's extremely complicated to use out-of-label drugs, and it's a responsibility of the director of the hospital. So you need to have to provide data from the literature, but you may use it, and you have to pay for it. It will not be reimbursed by the social security if it's like that. So in my hospital, as in many hospitals, when there is a commission deciding, from some data, if we decide, first of all, the treatment out of label, then I, as the director of the hospital, has to give my the green light to do that, and then we have to file specific reports for the health authorities, explaining what we are doing, because we need also to be covered in terms of insurance on that, and they may refuse. The problem is that if we have a lot of cases like that, it's extra cost for the hospital, because the patient is not going to pay. The patient never pays in France for that. So it's not the same situation as in other countries, as you can have an off-label, but the patient is okay to pay, and or the insurance company. In France, it's not at all this organization. So for us in France, it can be a problem. I have another question. Yes.
Sorry, if I may can make a comment, is like there are two sides of us, two challenges. One, to make sure we are not reinventing the wheel for each patient that might be similar to the prior one, even if we have one in a blue moon every 2 months, every 3 months, right? So we are learning from the from the past experience. And second point is more an access component, which is very particular to each country. In some of the countries, we might have a reimbursement in place already for this situation. In other countries, not, and it's very case by case discussion with the payers or the out of pocket money. So there are these two angles of two challenges of the same problem, I would say.
I have from my side two questions. One question is the integration of the clinical data and of the imaging data, because also you have some targets, but you will have a patient with brain metastasis or bone metastasis only, or with a lot of liver metastasis. So this is also, I think, information that are very important to collect also, because we know that depending on the extent of the disease, on the localization of the different metastases, some drugs may be more or less efficient, or should be combined with other treatments. So do you plan to do that? How do you embrace that? Do you plan to also enter response to treatment data from in kind of large data set, like RECIST?
Absolutely, Laura. Yeah. Laura, this is on the real world data stuff we constantly talk
absolutely.
about.
It's exactly that, and also extracting the information from multimodalities, and also from other type of data beyond genomics, right? And this is what we are talking about. This is also, whenever you are taking decision, also in a classical tumor board, no matter now what definition of a classical means, can be routine or advanced, or even advanced clinical research. We need different types of information, and some of information is more relevant in one situation or another. So yes, this is something which is ongoing, and we do recognize the need of taking information and triangulating the information coming from different modalities.
And context is so critical for variants. I mean, a variant really, in my mind, is meaningless without the clinical context and really the clinical picture, because what does it all mean, right? I just wanted to add that, because I think the context is very critical, the imaging, the clinical profile, etc.
And this was one of the questions that was what is the value to discuss about variants alone without the clinical information, and also without the therapeutic option clinically approved. But and also if it's post-treatment, post-radiation therapy, I think there is also some important impact on those variants. I have another question that is, the GAGO, garbage in, garbage out. When we collect all those data, and I know that it's sometimes when we go to the health authorities in France, defending a treatment or showing real life data, they always question the quality of the data that we have studied in the model, and on which we have based the model. So for that, I think that some of the models that you are presenting, yes, are based on FMI data or on standardized molecular data or immunohistochemistry data. How do you cope with that? Is it, or do you plan to enter data from other other molecular tests, for instance, or?
Absolutely. Well, on the Roche Information Solutions side, one of the key projects or tenets of what we're doing over there is around building a common, what we're calling kind of a data architecture or data lake, that's going to be able to, and I mean, we're in the process of doing this, to be able to take discrete elements from the EHRs and marry that information to stuff that's entered by the clinician as well, but marry that information to curated data from the outside, which would be guidelines, literature, drug labels, etc. And then to be able to so I think that the key piece here is you avoid the garbage in, garbage out when you start to structure and model your data, and you're able to normalize it in a way where you can look at it across the various data sources. So, I mean, that's the way I envision us tackling this problem of garbage in, garbage out. I don't know if you have anything to add, Laura.
No, it's absolutely the case, and also I would say I would add to that, starting with the right data, knowing that at the moment, what does it mean right data? Structured data. We do not have a big proportion of a structured data at this stage, and this is what we need. And this is also was one of the point of the final conclusion of the presentation, is that the molecular tumor board also as a way to prospectively structure the data.
Yes, data structuration is very important, and we see now when we participate to some studies or epidemiologic studies, retrospective studies on database, it's so important to have structured data to be sure that you are exploring correctly the parameters. We have one live question from Andrea Galabini, sorry. We can't hear you.
My question is very simple and comes from my experience in the world of imaging. And as a matter of fact, we tried to standardize response criteria in multiple myeloma by FDG PET-CT. And this could be a very useful tool, and also in the same time, it emphasized the limits of so-called molecular tumor burden in multiple myeloma, as we considered, for instance, the multiclonal step evolution of the tumor, which could bring out of detection of the minimal residual disease, moving from classical molecular biology. And as a matter of fact, we found some patterns of disease recurrence or progression escaping the classical control by molecular imaging. So it's a good field to sketch out a possible integration between imaging and molecular biology.
Absolutely. That's interesting.
That's obvious, sorry, but yeah.
Well, and,
it's a good point.
It's a great point, and it also brings forward this idea that we really need to be monitoring across, across the disease continuum, and not just think about this as, okay, we're going to pick a therapy and then we're done. This has to be, the tools that we build have to have an eye towards monitoring for minimum residual disease and the process along the way. And so that's something we're very keen on in Roche Information Solutions as well. Thank you.
Okay, thank you so much. We have other questions. So one question is, how would you see all these technologies integrated in clinical trials?
That's a good one.
Yeah, I don't think it's a solution fits all, or it can be in a staggered approach or in multi-step approach, right? First of all, we need to show that the integration of different technologies is right. The integration is done in a proper way, and it's working, and it gives the right responses that we know already in order to identify new answers, right? It's something which is ongoing. Doesn't mean we are still yet there. There are some clinical trials already which are including different technologies beyond genomic, and yeah, these are some things which are already ongoing.
Okay. And also the data, if the data from the clinical trials are captured with the tools, it could be also important because they are bringing informations also in the model. In fact, they are feeding the model, and could be also, in my opinion, important, even if it's more selected patient usually in clinical trials. So we have questions that are maybe more practical. About so it's from French, so it's RCP, but RCP is multidisciplinary tumor board. So for many clinicians, if we do a specific molecular tumor board, that's what I was starting to discuss with you previously. This add another multidisciplinary tumor board, so it could be very complicated to manage in their agenda. So what is your vision on that? What is your point of view? And there is another question, but that could be also answered together, is how could we involve more clinicians and also the private clinicians in the molecular tumor board? Because in France, as you probably know, we have the university hospital and the general hospitals, the comprehensive cancer centers. All those hospitals are academic, and then you have the private sector, and a lot of oncology also is in the different hospitals from the private sector.
I can take the first question. So on the question around the time and that it would take, and why would we want to add another meeting? Well, another molecular tumor board. I've seen it work really well, at least in other countries, where there is a upfront what is called maybe a curation meeting that happens among the bioinformaticians and the clinical geneticists and molecular biologists, and they really sort of put together the story, which then is taken to the oncology team, the medical oncologists, surgical oncologists, et cetera, in a much quicker, rapid meeting, 5 minutes per patient. But I mean, all that upfront work, which is necessary. And the hope is that clinical decision support tools can really help kind of make even the upfront curation work quicker. But the idea would be that this would then be taken to, the molecular tumor board, and these would be 5 minute per patient kind of discussions. I mean, I've seen it in action, and it's working. It just takes a lot of work up front by the clinical geneticists and bioinformaticians and molecular biologists. So I don't know if that's a model that you envision working in France, but I just wanted to highlight that.
True that we already show the data for the clinicians and predigest the data for the clinician happily, because sometimes it's very complicated, and especially when you start with the fusions, and it takes
Yes.
2 or 3 days to interpret everything, to go through the literature, for the international database. And yes, because I think it's important also to understand for the clinicians that it's not results that are arriving like that on a form from the machine saying this is this and that, or like when you receive a FMI result form, the raw data much more complicated, and it takes a lot of time to go through those data, check them, and then as I said, go through all the database, check the database for the variants, for the
and is it germline or somatic? Spending time going, do we think it's somatic even? Is it gain of function or loss of function? Those discussions are lengthy.
So
I didn't mean
one
to
of
cut
give
you
you
off.
a try for the second. I can give you the try for the second for the second questions regarding the private hospitals. I will start with the question now, maybe more a rhetorical one. When do we need a molecular tumor board, or when the molecular testing is done in the patient journey, and what patients are treated in their time in points are then the patients when they are captured in the private hospitals. So this is a question that I'm not necessarily having an answer, but to the best of my knowledge, an NGS or a comprehensive genomic testing or FMI or anything, it's done for patients which are the end of their journey, treatment journey, at the end of guidelines somehow. And in these cases, most of these patients, at least in different countries, I have some examples of Switzerland, Austria, and Germany and other countries where these patients are referred mainly to big academic centers. I don't know if it is the case in France the same way, but I'm very happy to understand that as well.
Yes, well, usually in private practice, the patient have simple genetic testing. So just, RAS, BRAF, and some of them can start to have targeted NGS. So that's true that when everything is negative, and they could refer those patients to other hospitals, but it's not always the case. Sometimes they treat them by chemo or other treatment, and sometimes they refer the case to screen, or they send us blogs to screen for clinical trials or for unusual alterations. What I was thinking, because that's what we do. Most of the cases, they are included in the organ tumor board, especially in my hospital with lung or with breast or with ovarian cancer, because in fact it's relatively simple. It's when we have strange cases or complicated cases that we will have specific meeting for that, and sometimes they are not only at our hospital level, but they can be regional level. So there are more specialized multidisciplinary tumor boards that can occur at, for instance, in lung, at a regional level, involving people from different hospitals. This could be something. I think that the pre-screening, the triage of the different patient files before the multidisciplinary tumor board is something that is important, because we know that before a multidisciplinary tumor board, so you will have all the data from pathology, from imaging, from the clinician, and then you will have clinical research technicians who are looking through those files to pre-screen the patient for the clinical trial. That's what we do in my hospital. They look at all the files before the multidisciplinary tumor board to see if some patient could enter clinical trials. So also there is already a pre-screening that is done by the clinical research team. But maybe it's because we are in a research hospital and in academic hospital with clinical research. So this molecular evaluation is already done for our patients also by that, but it's not done by everyone. And I hope that in the future we will have tools going through our structured data, triaging the patient already with all those data, triaging the patient for the different treatment option, classical option or clinical trials option. I think we have. Do we have again? Yes, well, it was one of the questions that I answered. In order to have the support of the people in charge of the tumor board, it's important to have tools that can be connected, plugged to existing tools to avoid data re-entry. Yes, this is a nightmare when you have to do data re-entry, but when you have structured data that can be collected by interconnected with other tools, I think that's what is important, and it should be a priority, I think, for many people. I don't think that we have more questions. I don't know if you want to add something, Laura or Tara.
No,
That's okay
I'm
for you?
very
No, I
fine.
just,
Thank
I
you very
know,
much. Thank you very much for inviting us. It's great discussion.
I enjoyed a lot this meeting. Oh, we have Tufik who wants to say something. Hello again, Tufik.
hi.
Hi,
Hi, Tufik.
Frederic. First of all, really big thanks to you, to Laura and Tara. This is really a great session. We know that we have 5 minutes to go, but I have really maybe a provocative question to you, Frederic. With regard to the tumor board, how is the patient involved in the decision? And whether you have any challenges that sometimes the patient discuss and also refuse any decision, a clinical decision coming from you guys?
Mm-hmm. So of course, sometimes we do everything and the patient refuse treatment. So it's usually when we have a patient refusing any treatment, we try to see we have an oral drug to offer the patient. And sometimes that's why we do molecular testing, saying, because sometimes for the patient, what is difficult is to go to the hospital every 2 weeks or every 3 weeks to get injection, or, to go from their home, especially if they have bone metastases, it's very painful to travel. So sometimes we will do molecular testing for that, to offer oral drug and some things that could be better for the patient. Then, if we have a patient who is refusing everything, refusing treatment, we cannot force the patient to be treated. And in fact, you are completely right. It's something that we should be asked to the patient before, and maybe sometimes it's better we don't know, because if the patient doesn't want anything. But as a medical doctor, I think we must do everything that is in our power, and use all the tools that are available to treat the patient. So that's why, even if we know that the patient is reluctant to be treated, we try to find something. Then there is another situation where we have a patient with late stage disease. Sometimes the patient is exhausted, but the family is very pushy, and the family is really putting a lot of pressure on the medical team, saying, can we try something? Can you look to some very rare alteration? We are ready to go to Paris or to Bordeaux or to Lyon, to another city, if there is a clinical trial, and everything. So here, this is where ethic committees are very important, because sometimes it's very important to have the patient point of view, and especially for the late lines, and sometimes the patient is okay, and sometimes the patient says, no, for me it's the end of the road, I don't want to go. And this is something that we will have to take into account in the future for those treatment, because sometimes we will find alterations, but the patient will have to travel sometimes to go to 2, 3, or 4 hours driving to another hospital to get a very innovative treatment. So you probably will have to settle there, to stay there for a while. And some patients, they don't want to do that. They are exhausted. So I think that you raised a very important point, is where do we work with the ethical committees, and where the patient is involved, and where the family is involved, and where at what moment do we have really to discuss what is the family wanting and what is the patient really wanting. And this is a situation that we encounter very frequently, in fact. And it's very complicated to manage, of course. If we don't have more questions, I want really to thank you very much, because it was a very, very interesting session. Thank you also for the participant and for all the questions that we had. And so now there is a break, and we reconvene at 4 p.m. And it's a session dedicated innovation in imaging, personalized healthcare, and value-based healthcare. And don't forget to fill the feedback questions that will appear on the screen. And thank you really very much. It was very, very interesting and promising.
Very nice.
Thank you.
Thank you very much.
Thank
Bye-bye.
you.
Bye-bye.
Bye.